This is a production of Cornell University.
The gate now. So the three research areas that my lab encompasses here is focused on crop nutritional quality, high throughput phenotyping, quantitative genetics, and genomics. A large part of my research is focused on maize, which we all love, but it is a global starchy staple crop, and that would greatly benefit from biofortification for development of nutrient-dense grain. And why I say this is when you focus on this map here, and this is a map of maize consumption by mean percentage of daily calories per capita, you can see countries in sub-Saharan Africa that are having to subsist very highly in terms of their caloric intake from maize. And this orange country right here is Zambia. And in looking at the nutritional profile of maize, it often does not provide adequate daily levels of essential nutrients that we need to survive as human beings and also feeding to animals. And the ones that we work on in my research group is iron and zinc, and then provitamin A, vitamin E, and the B vitamin complex. But as you see here, maize is great in terms of a vehicle for getting carbohydrates out to animals and human beings, but it is very depopulated in terms of nutritional quality. And so as an approach, you can have a plant biofortifying itself, so harnessing the genes and alleles that naturally occur in the population or through transgenesis, or you could also complement through plant breeding with chemical fortification, dietary diversification, or in a sense, giving vitamins to people. But one of the arguments that I would like to make is biofortification is targeted to rural smallholder farmers, it's sustainable, and it's renewable. But these three other approaches should be complementary to biofortification because there is no single panacea that can be used to overcome nutritional deficiencies in our growing world population. One of the major traits or phenotypes that we focus on in my lab is provitamin A. And if you look at the global prevalence of vitamin A deficiency in the world and see the map here of sub-Saharan Africa, and that overlays quite well with in terms of caloric intake of maize, daily calories, and also in these sub-Saharan Africa countries, people are subsisting on a lot of other crops such as cassava, which are in a sense low in vitamin content as well. And the impact of this, there are 7 million pregnant women, 127 million preschool-age children that are vitamin A deficient in the world. And the consequence also, vitamin A deficiency causes 600,000 early childhood deaths, blindness in 500,000 children each year. And all of this is preventable through improved nutritional food consumption. And so when breeding any crop, the first question you must ask yourself, is there natural variation for this particular phenotype? And in maize, there is. So if we're just looking at the gradient of yellow or light yellow to dark orange kernel color phenotypes in a world's collection of temperate and tropical maize throughout the world, if you look at the total carotenoid profile in the maize grain, beta-carotene, which has the highest provitamin A activity, only accounts for 6% of all those carotenoids. And I want to highlight that when we're talking about carotenoids in maize, they're mostly found in the endosperm. And when you break out that 6% slice here, on average, 1.7 microgram per gram are found in these maize lines, so that is not very much. And Harvest Plus, which is very much focused on biofortification of our important food crops, has worked with human nutritionists, and they highly recommend a tenfold increase as compared to like the average there in terms of this as a set breeding target. And this is a breeding target that people have been trying to reach, and some breeding programs have actually surpassed at this point in time. We also work on vitamin E in my lab as well. And in this case, I would kind of define vitamin E more as an insufficiency. And when you look at reports of insufficiency in the world, that would be in countries such as Brazil, the US, Tunisia, South Africa. But in the US, about 26 or 41% of individuals have plasma alpha tocopherol content in their plasma that is at a level that increases their risk for cardiovascular disease. And so although few reports exist for other developing nations, it is thought of that these concentrations are probably even lower and more widespread. And thinking about vitamin E and more so alpha tocopherol, which has the highest vitamin E activity in the human body and also animals, we see here of all the tocotrienols found in maize grain. Here we have alpha tocopherol in this shaded blue color right here, and that accounts for about 11%. And almost all of that alpha tocopherol is found in the embryo of the maize grain. And here we have a higher average as compared to beta-carotene 8.19. And if we do the calculations and working with human nutritionists, this is approximately close to a five-fold increase that we would have to have as a breeding target on average. And so when we look at where we were when we began this one project about in 2010, the first take home here is a lot of these compounds are all derived from a shared biosynthetic pathway, such as like the aromatic amino acid pathway and the GGP pathway here. So here we have an HGA, which is coming off and being used as a head group, and a Phytol DP, which is important for the tail group. And these, through the chemical action of the enzymes themselves, give rise to all the chemical variation for tocotrienols in maize grain. And these two genes right here, BTE1 and BTE4, had been, in our lab, had been found to be underlying large effect QTLs. And for the carotenoid traits here, we see the two genes LIKE and CRID-RB1, which have been published on previously in 2008 and 2010 out of the work here at Cornell and Ed's lab. And these two genes themselves have been used in the Sim and Harvest Plus breeding program to develop orange high provitamin A maize. So that seems to have worked very well. But these particular genes themselves are only capturing just a snapshot of the variation in the maize germplasm pool. So what we're trying to now do here is use large populations to try to map the locations of other QTLs genes in the maize genome. And here we have HapMap version 1 and version 2 that have been genotyped and imputed and projected in the maize NAM population here, and working with our colleagues at MSU and Dr. Dean Della Penna's lab, we've run HPLC and almost 20,000 HPLC runs to quantify the carotenoid content and composition, and the same for tocotrienol, vitamin E, tocotrienol content and composition. And expression profiling has also been performed as well on all of the like 26 NAM parents, and also at 6 developmental stages post-pollination times 40,000 genes. So in a sense, we want to really understand which genes are turned off over the developmental program of the kernel itself, and to also put the QTL on a time axis. And to carry out this research in the maize NAM population, what we first do is what is referred to as a joint linkage analysis. And here we're identifying the QTLs in the maize genome. So you can think of the maize NAM population as a map within a map. And here we're just using the recent recombination that has happened in the construction of the maize NAM population. So in a sense, we're just identifying the chromosomal segments in the genome that have that QTL. And with the GWAS, which now we're using a higher quantity of SNPs, here what we're now using is the ancient recombination. And so this is through the evolutionary history of maize, and you can see the LD blocks compared to the recent recombination map are much smaller. So this is where we're going to receive the higher resolution for trying to finally map and hopefully identify the causal genes that are controlling variation for these important nutritional grain traits. And so in our work, what we've found here, if we just focus on the carotenoid traits first, we found that essentially 10 a priori pathway and 2 novel genes mostly underlie the natural variation for these carotenoid traits in maize grain. And just to walk you through this, DXS2 and 3, so these 2 genes were involved in the parental tail synthesis. 3 genes upstream of the pathway branch point right here, PSY1, PDS1 in mutans right here. And then these 4 genes within the core pathway right here, LIKE, LUT1, CRID-RB1, and ZEP1. And we reconfirmed LIKE and CRID-RB1, which had been previously used in the CIMMYT Harvest Plus breeding program. And we also identified a carotenoid cleavage enzyme, which essentially is turning over the carotenoid compounds and chewing them up, which is definitely not what you want. And so when we dissect this even further, essentially about 6 a priori pathway genes that are what we refer to as expression QTL are explaining the majority of the phenotypic variation for these traits. And this is LIKE, PSY1, ZEP1, CCD1, DXS2, and CRID-RB1. And in a sense, these variants that are associating with the phenotype are cis, and it's explaining the fact that they are expression QTL. And then the other genes that were hit do not appear to be expression QTL. So perhaps that the actual SNPs or the causative variants are changing the activity of the enzyme itself. That would be like LUT1, DXS3, and mutants and PDS1. And then the 2 novel genes that we hit here, one is an uncharacterized protein, so you can obviously just hypothesize what you think it may be because it's uncharacterized. That's a great thing about candidate genes in this context. And then the other one was a MIB transcription factor. I would like to point out that these 2 novel genes here, though, were only associated with total carotenoids. So with this maize NAM population of 5,000 recombinant inbred lines, we were able to really dissect down to the gene level the largest effect QTL. And when you focus on this orange part, so this is the amount of like phenotypic variants explained by a priori genes. The gray part is the amount explained by those 2 genes that are novel. And these are unresolved loci. In a sense, we don't really know what those causal genes are. So by seeing this, you could probably be saying, well, why'd you have to use the maize NAM population? You could have just done a candidate gene approach. What we didn't realize at the time is that the known a priori candidate genes would explain the majority of the variation, but for the genes that are unresolved, they explain about 1% or less of the variation. And in those regions of the maize genome, there aren't any a priori candidate genes. So those are things that we can certainly follow up on, but in a breeding context, they don't explain that much of the variation. And so now when you think about breeding for beta-carotene, which is the compound that has the highest provitamin A activity found in the maize grain, these 5 genes here are the ones that you would focus on: DXS2, PSY1, LIKE, and CRID-RB1. And so you can think of very much as this is just reduced to a combinatorial breeding approach, and you're just trying to figure out the best allele combination to put together in your breeding program so that you can calibrate the composition and the total for the type of carotenoid phenotype in your grain. And just these two genes that we have just talked about previously that have been used in the Harvest Plus breeding program, they have been used to release new hybrids in Zambia. And this is an outgrowth of the orange high provitamin A maize. And some work that was published about 5 years ago showed that this orange high provitamin A maize was as effective as providing vitamins to Zambian children that had low levels of vitamin A. So it isn't just that we were able to select on the genes themselves, but what was shown by nutritionists is that this product, this orange high provitamin A, is as effective as giving vitamins to the children. So I think this is one of the rare cases where you work closely with like the growers and the at-risk population, and you understand their needs, and that's something that Harvest Plus does very well, so that after maybe 8 to 10 years of developing a new hybrid or a new inbred line, people will actually use it. And some of the work now that we're focused on is really on deployment of these genes, and then understanding how well these hybrids are performing under heat and drought stress. And Christine, a former PhD in my lab, worked for a year in Zimbabwe with CIMMYT. And here is one of the field days that they had, where they were showing to the public these new released orange hybrid provitamin A inbred lines, hybrids, and then showing people how they look after being cooked, and then they could like taste them as well. Here's an image from her drought test that she was doing, and where about 300 hybrids that were bred to be high in provitamin A were tested under heat and drought stress. And we have all of this seed now in our cold chamber. We'll be analyzing it as well to see how the composition of that grain was perturbed over time from the heat and drought stress. So moving on now to vitamin E, this is a nice contrast here, because here we found that a priori, 8, that is a priori pathway genes, and then 6 novel genes explain about 60% of that natural variation for tocotrienols traits in maize grain. And here we have DXS2, DXS3, and if you recall, those were hit for carotenoids as well. So the RADH2, HPPD1, SDS, BTE3, HGD1, BTE3, and BTE4. So the majority of the phenotypic variation, though, is explained by a priori genes for tocotrienols, and tocotrienol traits have high antioxidant capacity, and that would be such as the gene HGGT1. But we found that novel genes are mostly controlling the variation for tocopherols. So if you look down in this part of the table here, so this would be like these snare proteins, the fibrillin, this transferase protein, and PHD gene here. And working with Dean Della Penna's lab in Arabidopsis, dare I say, we've been able to confirm that some of these genes do have a contribution to this pathway. So that's really nice, because we've been able to take the gene from maize, put it in a line of Arabidopsis that no longer has this gene functional, to show the confirmation and recapitulation of the phenotype. The 2 genes that I would like to focus more on now are the protochlorophyllide reductases, POR1 and POR2. And this was a real surprise to us that we found these genes to have a very large effect. And these 2 genes, they're homologs in the maize genome, explain almost all of the variants for total tocopherols that we found. So why this is intriguing is because these 2 genes are involved in the chlorophyll biosynthetic pathway. So here we have protochlorophyllide A. The POR gene is doing a reducing reaction here. Then we have the chlorophyllide A, which lacks a phytol tail. And then that's added through the GG here, geranol, and then we have the chlorophyll A and B right here. And so this is where we begin to hypothesize what may be going on. We did analyze the embryo and the endosperm of these developing maize kernels, and we found trace amounts of chlorophylls that are present in the maize embryo. Just to remind you, the maize grain is non-green, non-photosynthetic tissue. So why is chlorophyll in it? Okay, and so when you look here, this is days after pollination here on this x-axis, 16 to 36 days after pollination. And this is from the embryos, and this is average across the 25 NAM parents here. And on the y-axis is the log concentration of each of these compounds. Tocopherols here, you can see is increasing over time, but you do actually see chlorophyll A here, chlorophyll B, and then a precursor of chlorophyllide A. And this would be more related to a degradation product without the magnesium, pheophytin A. And this is just to remind you what that the Phytol group here is important for defining chlorophyll A versus chlorophyllide A right here. But when you think about the amount of chlorophyll that's found in the embryo versus the leaf, the chlorophyll to tocopherol molar ratio is 1 to 800 in the embryo. Thus, we feel that the chlorophyll degradation is not sufficient as a source of phytol to generate the tocopherols in the developing embryo of maize grain. So what we think is happening here is actually a cycle. So we hypothesize that the phytol for tocopherols in embryos is most likely provided through the chlorophyll biosynthetic pathway. So first, just to draw your attention to this plot here, this is days after pollination again. And on the x-axis and the correlation of each of these different metabolites with the total amount of tocopherol here. And here we see a strong correlation over the developmental time period for chlorophyll A, for also chlorophyllide A and chlorophyll B, but we do not see a strong correlation at all. And in fact, it is very weak for pheophytin A. So this reaction right here involving these pores is a strongly light-dependent reaction. And so what we think is happening here is that the Phytol group is somehow being cleaved from the chlorophyll A, and that it's being used by VTE5 in the Phytol salvaging pathway, and that is what's leading to tocopherols. And here, if the degradation of the Phytol was very important and very strong, we would think and hypothesize that the pheophytin A signal here would be very strong, but we're not seeing that at all. And so for this cycle to actually happen, we have hypothesized that there are at least one or two more enzymes here to cause this cycle to actually turn. And we have some leads, and we've cloned genes now that we think are filling out this pathway part right here. One other thing is that we have tested out in this field, in the field season this year and also last year, since this is a strongly light-dependent reaction, what we did is after pollination, it was about 5 to 10 days after pollination, so we have our pollination bag on the ear. We have these foil hats that we're putting on the ear, so no light can penetrate the ear at all. And then after, comes October and the first snowfall, we go out there and harvest, and then we take off those foil hats. We make sure that there was no penetration of light, and then we analyze the samples with the HPLC. And so what we found here to test this hypothesis is we think if it is an important reaction involving these pore genes, that if you block out the light, then the amount of tocopherols should decrease as well. And that's what we saw in the first year, and we're just reconfirming this year as well. And so to complement that, we've also been doing CRISPR of these two pore genes as well. But as you would expect, if you CRISPR two important genes in the chlorophyll biosynthetic pathway, you would expect for the CRISPRed plants to be very pale and very green. So we are treating these plants as they are our own children, and we're trying to ensure that we actually get seed from them, and hopefully we'll be analyzing those by HPLC as well this coming spring. So that's our hypothesis that we're trying to confirm now with the chlorophyll cycle. So the largest effect QTL for the tocotrienols pathway has technically been resolved to the gene level and could be deployed in breeding programs. But as you see here, there's still a fair amount of unresolved QTL right now, and this is what we're following up on our PGRP pathway grant right now. And this work is being carried out by Di Wu and Jae-Woo Lee in our lab right now. And Matt Passaggio has been talking about this for about the past 4 years. We are trying to leverage all of our investment in understanding the genetic architecture of carotenoids and tocotrienols, so provitamin A, vitamin E, and the important xanthophylls, lutein and zeaxanthin, to improve your sweet corn. I know it's like winter and there's snow on the ground, but think about eating more highly nutritious corn, of course, covered in butter in the coming summers. So now we have done close to about 200 crosses, and we now have identified important genes that are also key players, such as LIKE, CRIDRB1, HGGT1, VTE1, and VTE4. And when you think about, you can have these important haplotypes fixed as your cofactors in your GS models, and then you can have the polygene effect capturing that residual variance. And in fact, the prediction ability for these phenotypes can be quite high in our breeding populations. So I, in fact, do have a breeding program, even though it's hard to believe sometimes. When I find time, we're actually out in the field, and it can be some very tasty phenotyping if you ever want to come join us in terms of quality traits. And so now I'm going to begin to switch gears to some of the high-throughput phenotyping work that we've also been doing in my lab over the past 10 years. And this was driven out by having some early discussions when I was doing my PhD here with John Luke, when I was still trying to figure out what is genomic selection. So I would chat with John Luke. He would be like, it's, you genotype and phenotype a population, this training population, you train a GS model, and then ideally you genotype your breeding material and try to predict what those phenotypes actually are. But even though you're doing that, you still need highly accurate phenotypes, and you still have to be doing phenotypes as you retrain the models after, say, every 3 or 4 cycles as the allele frequencies are changing. And why I began working on this, because in Arizona, I was very focused on trying to improve the heat and drought stress tolerance of cotton. And as with new nutrition, I think about children and the vitamins they're not receiving. But in this case, during the early part of the 2010 through 2012, there had been repeated droughts, including in Texas here in 2012, where a humble cotton farmer can just lose their entire crop from drought. And so what I quickly began realizing is we needed a very effective way to do high-throughput phenotyping for heat and drought tolerance in the field. And how we began doing this when I was working at the USDA was having this high clearance tractor right here to phenotype canopy temperature, NDVI, which is basically a vegetation index, doing like chlorophyll content, if you will, biomass, and then height. And then we were testing this on a recombinant inbred line population of cotton, close to 100 recombinant inbred lines. We had also phenotyped agronomic traits at the end of the season with a combine or cotton picker going through the field and various yield components, fiber quality traits, and physiological leaf traits, epsizic acid. And the epsizic acid concentration was here at Cornell, thanks to Tim Setter. And we were doing all this under drip irrigation, so we can have a highly managed stress environment. We would calculate how long and how much of the irrigation should be applied for these 2 wet and dry regimes right here. And we were able to do this phenotyping over the entire growing season. So say we would phenotype every Thursday, and then we would do at 4 time points within that single day over the whole growing season. So in a sense, we really wanted to understand how is this particular population responding to the imposed heat and drought stress over time, from at flowering to at the end of harvest, when a cotton picker would drive through the field and quantify yield. And so what we were able to do this is essentially paint the genome to see what QTL are turned on and off as a result from the dry stress, the wet well water control treatment. And on the x-axis here is the year. So we did this over 3 years, 2010, 2011, 2012. And this x-axis here is the position along the genomes. Each of these are a chromosome. And so what you can easily see here in this very almost crazy painting, if you will, is that you can see the time points when certain QTL are turned on and how long that they're turned on for. And then you can kind of see if they're turned on between years and across years as well. And the red are alleles that are increasing the canopy temperature, and the blue are alleles that are decreasing the canopy temperature. And this work was carried out by a former postdoc who's now at the University of Arizona as a new professor. So here we have a nice candidate gene, ABA, but this is a large genomic interval here. But with a candidate gene, you can always hypothesize what may be happening. And why ABA may be important is, in this context, because when we had mapped the cotton leaf ABA content, the QTL mapped to the same genomic location as this QTL that was turned on very strongly for canopy temperature. Why that is important is as the stomata are open, transpiration is high, and as ABA is expressed in the leaf and becomes to a high concentration, the actual stomata close and transpiration decreases. So in effect, this could very much have be somehow linked to changes in the canopy temperature in terms of variation in this cotton recombinant inbred line population. But that is just indirect phenotyping of a trait such as, really what you're trying to get at is the amount of water in a leaf. So what we've been doing over the past 2 years, working with Weijin Liu in my lab and Piyush, John, and Abe Strook's lab, is developing these nanosensors that we call AquaDust to infiltrate into the leaves to more directly quantify water potential. So how this is working is have these like small fluorophore particles that are hooked onto these particles, and when you infiltrate into the actual leaf, they will expand and contract based on the leaf water potential. So if the water potential is very low, that means the plant is undergoing stress, and so then you would have shrinkage of these nanoparticles, and then these fret particles would begin to interact with each other, and then you would have a high fluorescent signal. And we also did validation, and we were showing here in this curve, so this is like water potential from a pressure chamber. You've also maybe heard it referred to as a pressure bomb. So all of this is an actual work that you actually have to like cut the leaf off and put it in the pressure bomb and then quantify this. But on the y-axis here is the fret efficiency of those particles, and you see it's following this line very well. So what this gave us confidence in telling us that we are able to directly measure leaf water potential in the leaf using these nanoparticles. And this is a confocal image here showing the distribution of these nanoparticles in the leaf themselves. And you can see that the AquaDust is residing in the apoplastic region, and really what it's doing is providing a measure of the local water potential in that leaf. So this is really, I think, one of the first steps to really begin high throughput phenotyping of water potential and its direct quantification of water potential. So we wouldn't have to do it through canopy temperature. We wouldn't have to do it with a pressure bomb. This is injecting particles in the leaf. So the next part is going to be about biotic stress. And this is what happens when you hang out with Rebecca Nelson. You begin working on northern leaf blight. So as I've become to learn and actually call these lesions beautiful, because when they're not showing up in our field in the season, it is not beautiful, as we have found out this past summer. So the fungus that causes these brown necrotic lesions, these lesions can begin, to grow and coalesce over time. And unfortunately, NLB disease has actually beginning to increase over time since 2012. And it can be high as a 1.9 billion economic loss. And that was the case in 2015, considering US and Ontario. So this is really a disease on the rise. And one of the challenges here, and some of this work really began with Jesse Poland when he was here as a PhD student, same time is really how do you rapidly visually assess NLB disease presence or severity, which is a quantitative phenotype out in the field, and it often is very slow and imprecise. So here we have the case where this is our NLB true positive, this is our NLB true negative. But even just trying to see these lesions by eye, assuming you're able to find a positive one, is very slow as you walk through the field. And then quantitatively is also very challenging when you're trying to do severity, which is in a sense disease-leafed area. Because here, this is the case of mean intra-rater radiability, repeatability of only a 0.76. So this is like comparing the same repeater to themselves, going from say in week 10 to week 12, and then seeing how close their ratings relate. And then the inter-rater repeatability is actually even lower. So this is not only slow, but it is imprecise when you're trying to do it quantitatively for disease-leafed area. So the idea that we had, and this is where hanging out with computer scientists and engineers is very sometimes intimidating to learn their language and understand what they're talking about at first. But as soon as you get out your comfort zone and you realize that they can do so much more to help your research, then I'm hanging out with them all the time and having conversations with them every week. So the idea was, and this is what a lot of people refer to as machine learning, deep learning. And this is us using these CNNs. And you can just think about using a CNN as converting a pixel into a logistic unit, 1, 0, presence, absence of a lesion. And what this is essentially doing is where we're applying these filters to detect features, and that's what is referred to as a convolution. And then the pooling is really simplifying the layers. You're trying to condense this information to allow more filters to be applied. And then you're extracting these multiple layers of the non-feature. And then almost like an ensemble classifier is combining all these features to make a prediction at the end. Either the lesion is present or it's not. And so what we've been doing, and this has been carried out by our wonderful pilot, Nick Hasmar, in the lab, is using the UAVs to really speed up the large-scale disease detection in the field. And here's one of those UAV images right here of some really nice lesions on these plants. And working with Hod's group at Columbia University, Harvey Wu and Chad Deschamps, and here at Cornell, we've been able to do highly accurate NLB lesion detection here. So after we have our trained CNNs, we've been able to achieve an accuracy of almost 98% with a holdout test. So we were actually able to detect these lesions, the presence of absence of them, very well out in the field. But we really want to do severity. We want to make Rebecca Nelson happy. And so this is what we've been trying to do over the past 2 years, is make her happy. And we're getting closer. So the great people at Facebook have developed what is called a masked regional CNN, short for masked-R CNN. So this is what we began doing, and this work is led by Ethan Stewart in our lab. And so you can see a nice lab photo right here of us all happy and smiley. And then we can run that masked-R CNN here, and then it's running these classifiers. So the idea here is you're conducting object detection by drawing these bounding boxes around each class, and then you're doing like a segmentation algorithm on each of these bounding boxes to classify a pixel as an object. So what Ethan has been doing is really trying to carry this forward into the plant world from the Facebook world. And so here we have an image of a disease lesion here, and here is where it's been outlined by a human being. And this is where the masked-R CNN would predict that there's one lesion here, and then drawing the area around that one lesion. And then the IOU is the overlap in that area. So this one actually looks fairly well, but there are cases when it does not look very well. And this looks like we have a piece of an anther or a tassel here, occluding this lesion right here, which is where a human drew it very nicely here and drew a part of this lesion here. But as you see, this piece of a tassel is kind of confounding the AI. So to overcome that, we just need a larger training set. And Ethan may have to drink more coffee. So after that, so with the UAVs, what we've begun working now within an Ed's group here is Cornell, and is trying to really understand how you use robotics for phenotyping and incorporate them into plant breeding programs. And this is with Girish Chowdhury's group at the University of Illinois, Urbana-Champaign, and this is on our Terra MEP project funded by the RPE. So the intent was to develop this for phenotyping biomass sorghum over time. And so what we're now developing with our lab is a comparative HAP map, and that's trying to integrate the evolutionary histories of maize and sorghum to improve the predictions of these complex traits in sorghum. And Jonathan here is here for 1 year from Brazil. What he's beginning to do is develop these Bayesian models that can use what is referred to as forward training cross-validation, where you can say have a robot going through the field, but then you're incorporating these time points early in the season as part of your training set to try to predict a phenotype at the end of the season. So in this case, this is height, and we're using days after planting, 30 and 45 days, to train the actual Bayesian model. We're using that information to predict height at the end of the season. And that, as we found, been able to increase our prediction accuracy. So when you think about a high-throughput phenotyping approach, can you begin to incorporate phenotypes early in the season to improve your accuracy so you can predict something such as height or yield more accurately at the end of the season? So you can think of this as saving time, then you would know early on what crosses to make early in the season prior to flowering. And as Jonathan has also shown, at certain later weeks after planting, there isn't much changing in the rank order of those plants for height. And having a robot makes you very happy, as I have found. And Ed would probably attest to that as well. So what we're really beginning to think about and do here is trying to integrate these unmanned ground vehicles, these unmanned aerial vehicles, to really tandemly phenotype NLB disease presence and severity from the understory to the canopy. Because I have now learned that the NLB disease starts at the understory and it works its way up. If you're seeing it from the air, it actually may be too late. So what we're now trying to work on is do an in-sync phenotyping approach where we're trying to integrate the information from the ground rovers and the UAV to really understand the rate of disease progression over the whole growing season. And with that in mind, and this is where hanging out with computer scientists can be very fun, we're really thinking about now a concept of a software-defined farm, where you are using the TV white space and a software called FarmBeats, and using that to eventually do end-to-end image processing nearly in real time as you're collecting the UAV data or from the ground rover to really understand how diseases are breaking out and how you would better manage your crop. And this is under the umbrella of CETA, and this is with my colleague Hakeem in computer science. So this is kind of where we are beginning to see and take high-throughput phenotyping, where it was once just thought of maybe we would just focus on one trait, but it's really now beginning to think, how do we fully integrate this in a breeding program, but how do we employ it to also better manage our crop at the farm level, and how do we sense and react at the same time? Because then you can think about having like a UAV or a ground rover with the highly concentrated canisters of like fungicide or a pesticide that can have variable application through the field over time. And that's where to have that actually to be possible would require something as a software-defined farm. That there's a lot of people to thank and funding agencies. For the carotenoid work would be at MSU and Purdue University, and here at Cornell with Ed's group. It's like the CNN disease work would be Hod's group at Columbia University, Becca's group here at Cornell, and for the EarthSense, that would be our rover, and then the phenotyping early work was involving colleagues at the University of Arizona and USDA ALARC. And unfortunately, I did not have enough time to talk about the other 20 projects that we have going on in the lab, because I didn't want this presentation to be just one slide for each project, but we're also doing some, I think, some great work in oat and cassava as well, and other projects in maize. But unfortunately, I just didn't have the time to go do a fast forward through all those projects. I thank you very much for your time.
Mike will take questions.
Mark.
Is there a negative correlation between vitamin E content and provitamin A content?
No, that's actually something we looked at very early on. And so DXS2 and DXS3 are kind of the only shared, I'll just call them precursor genes. And there isn't that much pleiotropy as we like previously thought. One thing, and Harvest Plus is probably going to begin hopefully breeding for this, is that since some of the tocotrienols are very strong antioxidants, that you can breed for them at the same time to help prevent the breakdown of provitamin A in the maize grain. And that has been shown in sorghum transgenically by the Pioneer group, but I actually think we can do that through breeding in maize as well. Great question. Yes, Susan.
I'm trying to process the idea that chlorophyll in the, if I understood it, it's in the embryo.
Yes, correct.
I'm just curious how light gets to the embryo within the kernel on the cob through the husk. And I've been I'm sorry, it must be obvious to everyone else. It's not obvious.
There is a very low level of light that can penetrate the husk leaves. We have found, but how much light is needed, I don't think it requires that much, but that is something that we're trying to figure out and follow up on. So we have also done, I guess, the counterpart of our test, you know, how we had foil on the cobs. We've also had ears where we took off almost all of the husk leaves, and our hypothesis is that the tocopherols should go up even higher. And that's, well, I hope to have the answer to you in like a couple months.
And is it a particular wavelength of light that penetrates maybe?
Oh, that's a good question. I do not know. Yeah, that's a great question. It's quite possible since it is going through like pale green husk leaves. Yeah, that's very possible. Yeah. Yes. Jeff.
You mentioned using the evolutionary history of maize and sorghum. What did you exactly mean by that? Polycloning? Is that what's in or what?
So, okay, so that's what I get for bringing that up. Okay. That's a great question. So what we're actually creating here is a comparative hap map. And so you can think about the diversity of sorghum, right? Pi, right? So what we're doing is using a machine learning model where we have all the different sorghum popgen parameters such as row, you know, other recombination rates, and, you know, other genomic features. But what we found is as we look at the syntenic regions between maize and sorghum, we're, say, getting pi and gene expression from maize as well, we're able to better predict the diversity patterns of sorghum using that information. So I think if this is the case where you are able to harness the syntenic regions that are more stable and shared, and so the idea is perhaps there'll be some of the shared, if you will, controlled of complex trait variation at the same time. And we've been playing around with this in maize and rice for the control of, say, like iron and zinc variation. It's actually kind of turning out to be very cool and very powerful, where we have orthology between the two. And then we're like, okay, there's a GWAS hit here, there's a GWAS hit here. And then we're able to kind of tease that out by just having like rice and maize. And so you can kind of think of it as the same way when you're using maize and sorghum as well. Yeah.
Other questions?
Sorry, Jim, I can't hear.
Phenotyping.
Yeah.
So for the lesions and the cysts distinguish?
Sorry, I like have to walk all the way back here. I totally can't hear you. Sorry. Yeah, that's a good question.
Repeat the question.
So the question was, for the NLB lesions, can you distinguish them from other abiotic stresses? And so we've actually, I guess, taken the easy route where we've just been focusing on lesions, but we know all those lesions are just caused by the one pathogen, right? I think as we begin to expand out and have this to be something that's more transferable and more valuable, you need to think about having the test of when there's like multiple diseases present and in the combination of abiotic stress as well. But the thing is, though, and the thing you have to keep in mind, at this present time, the humans are training the AIs. So if the human can't distinguish the 2 or the 3 or the 4, then it's going to be very hard to train the AIs. Sure, over time, as AIs train themselves, they may be able to figure it out, but we're not quite there yet. Yeah, good question.
If there are no other questions, let's thank Mike.
Thanks.
This has been a production of Cornell University, on the web at cornell.edu.